I'm 15 years old and in high school in Los Angeles. I love video games, exploring mechanics, and I also have depression, anxiety, and ADHD. From when I was a kid, I was always bright and intuitive. Under all of that, though, I was fighting to deal with depression and anxiety. Depression has always been near me, lurking, trying to make my life hell. Often, I really never want to do work, or even get up, because I really don't see a point in doing any of it. In 7th grade, my anxiety and depression was on an all-time high. I was so stressed that I started to cut. Later that year, I drank 3 bottles of beer in 20 minutes and nearly died of alcohol poisoning. I do not want anybody to ever have to go through that. ADHD basically makes my mind listen to 30 songs at once, trying to find just one specific song, all while trying to find a needle in an ocean of hay. ADHD also makes it very difficult to focus and not zone out in class. I take meds for it, but even with the meds, it can be very difficult to focus. This makes learning at school very challenging, and I often think that I am incapable no matter what I do. According to the National Institute of Health, nearly 1 in 3 of all adolescents aged 13 to 18 will experience an anxiety disorder. These numbers have been rising steadily between 2007 and 2012. Anxiety disorders in children and teens went up to 20%. About 20% of all teens experience depression before they reach adulthood. Between 10 and 15% suffer from symptoms at any one time. Rising anxiety and depression contribute to rising suicides by teens. Suicide is the second leading cause of death for college-age youth and ages 12 through 18. More teenagers and young adults die from suicide than from cancer, heart disease, AIDS, birth defects, strokes, pneumonia, influenza, and chronic lung disease, all combined. My family and I have tried all kinds of things to make things easier for me, and yet, every day is still a struggle. Recently, in my biology class, I was introduced to genetic therapy. Genetic therapy is a way that scientists can manipulate genes to treat or prevent diseases. Vectors are virus cells that have had their reproductive and harmful genetic strands extracted from them. Once the harmful and reproductive properties are out of the way, all the virus can do is safely transport and inject a corrected gene strand into a cell. The virus invades the cell and replaces the mutated gene with the corrected one and then just dies off. Scientists use a process called Crispr to help identify the targeted gene strands within millions of gene strands. CRISPR is a bacteria that can take specific gene strands and can store them. CRISPR is kind of like a fingerprint scanner. I can recognize a certain strand of DNA and treat it like a virus to cut it off and prevent future attacks. If you know a DNA sequence, you can use CRISPR to change it. That's why it's so amazing. Some people argue that gene therapy is dangerous and immoral. There's another type of genetic modification called germline editing. It uses CRISPR to make changes in the genome of an unborn embryo, which some people say is unethical because of the drastic repercussions if the procedure goes wrong. Germline editing will affect all generations after the gene is originally edited. The result of germline editing going wrong would result in side effects that could be unknown until a baby is born and now has to live out their life with that change or side effect that they did not ask for. There have also been accidents, which some people use to say that genetic engineering is bad. In a 1990s accident, Jesse Gelsinger's OTCD, a disease whose lying undercause is a mutation in the X chromosome, resulted in his death. OTCD prevents the breakdown and excretion of ammonia, which affects the whole central nervous system and especially the liver. Researchers at the University of Pennsylvania in Philadelphia injected him with working copies of the gene that had been attached to an adenovirus, a type of cold virus. The virus, altered to be harmless, would infect the patient's liver cells and integrate the added gene into their chromosomal DNA. However, he died. History has also shown us how gene therapy could be used in an evil way. An example of this is selective breeding, like the eugenics movement that started in 1883 from the ideas of Francis Galton. Put simply, eugenics is selective breeding to create the perfect human race. The methods that were used consist of preventing unfit people from reproducing by sterilization. Most of the time, sterilization was against the will of the person, but in extreme cases result in euthanasia. The primary targets of the eugenics movement were the poor, ill, and people of color. Eugenics started to lose their popularity during World War II, when Adolf Hitler used it to justify killing and sterilizing people unfit to reproduce in the tens of thousands. Some people use science fiction books like Brave New World by Aldous Huxley as evidence of how gene therapy is wrong. Brave New World is placed in a future society where emotions and individuality are conditioned out of children when they are young. I do not believe in eugenics and using gene therapy to acquire the perfect race. However, I do believe that gene therapy can be used responsibly to combat diseases like sickle cell anemia, diabetes, depression, and anxiety. An example of how gene therapy can help is the Michigan State University research project, where 5 dogs who were losing their eyesight went through gene therapy, and their eyesight was 100% restored. All 5 of them got their eyesight back with no repercussions. Using Crispr, scientists at Casey Eye Institute in Portland, Oregon, have injected a vector into a patient to treat a rare genetic form of blindness. Current gene therapy experiments are proving very positive results at high success rates, which are still improving. With the current pandemic of coronavirus ravaging without a cure, researchers at 2 Harvard-affiliated hospitals are adapting a proven form of gene therapy to develop a coronavirus vaccine, which they expect to test in people later this year. This discovery could prevent future pandemic shutdowns. Depression, anxiety, and ADHD are diseases where the 5-HTT-LPR region of the brain is affected, especially the SLC6A4 gene codes for the transport of serotonin, the chemical in the brain that relays signals from one part of the brain to another. Low levels of serotonin have been shown to cause depression and other psychiatric conditions. Some people have long and others have short alleles in the gene. People who have long alleles have a higher serotonin transporter, which makes it less likely for them to have psychiatric disorders. If the SLC6A4 gene could be manipulated, then serotonin levels can rise and depression and anxiety can be relieved where medicine does not work. So, from all these struggles that I've worked through, I've gotten to know depression and anxiety very well. I know how much it can affect someone and limit them. And knowing that there is a possibility to completely eradicate both of them is something that causes me to not sleep well until that is becoming reality. I want gene therapy to be used with integrity and caution to help everyone have an equal start. No physical or mental illness. All advances in modern history have had risks, and so does gene therapy. I believe that the risks that gene therapy presents can be worth endeavoring into. How many teens can we save from premature death due to suicide caused by depression and anxiety? Would you let all of the miraculous opportunities be oppressed by risks that are standard in the medical field? Thank you for your time. She laughed.
Hudson, TikTok, Part 2? Take 3.